Hello everyone, thank you for checking out my poster presentation. My name is Karsten Rinas. I'm a PhD student in the biology department at the University of Waterloo, and the title of my poster I present is A Smooth Bootstrap Method for Reliable Estimation of Type 2 Errors of RNA-seq Data. So why are Type 2 errors interesting? The statistical analysis of RNA-seq data can be challenging because of many possible biases introduced during sample preparation. Type 1 errors for detecting differentially expressed genes are well studied. However, type 2 errors are not, but a low type 2 error rate can improve the confidence in detected changes or the lack of it in experiments. Other proposed methods use a simulation method to calculate type 2 errors, which requires a lot of assumptions about the sample distribution, whereas Bootstrap needs very little knowledge about the actual sample distribution. Bootstrap refers to sampling method with replacement, and the here used smooth bootstrap is an extension to avoid recurrence of values from discrete data. Basically, the values get sampled from a smooth kernel density distribution instead of the input data. You can see the univariate kernel estimator in equation 1. As a kernel, we used a Gaussian distribution, and the smoothing parameter of bandwidth was estimated as described by D. Scott. To test our method, we used two quite different data sets. One dataset was derived from a cell line treated with two similar monoclonal antibodies and therefore contained three groups: control, treatment 1, and treatment 2. The PCA plot in figure 1a at the bottom left shows the low level of in-group variance but separate clustering of the groups from each other. The dataset 2 is from the TCGA-BRCA project and represents human samples from breast cancer stage 2B or 3A. The data has over 80 replicates per condition and shows very high in-group variance but very low between-group variance, as you can see in the PCA plot in figure 1b. For the workflow, as you can see in figure 2, for each dataset or subgroups with smaller replicate numbers, the read count output of HT-seq was imported to start the error detection. Then, the fold changes of the means were adjusted to the test effect size. Next, the smooth bootstrap method was performed on the data with 100 iterations, and then the genes were tested for significant differential expression with edge R. The final step is the error detection by calculating the type 2 error or power of each gene by estimating the ratio of how often the p-value was below the alpha threshold for the 100 iteration. An example power distribution of all genes is shown at the bottom right of the figure. The results section covers the figures 3, 4, and 5. The power for each dataset was calculated for various replicate numbers, effect sizes, and FDR thresholds. For the cell line treated with monoclonal antibodies, we compared the control group against treatment 1 and treatment 1 against treatment 2. For the TCGA-BRCA project, we compared the stage 2B against the stage 3A cancer samples. Figure 3 shows the average power for each set of parameters. On the left side are the power curves for data derived from the treated cell line, and on the right side are the results for the TCGA-BRCA project. The plots clearly show the constant increase of the power with increased replicate number for all comparisons. And with increased effect size, the power exceeds the common power threshold of 80% already for very low replicate numbers. For the drug-treated cell line data, the power is over 80% for almost all replicate numbers for an effect size of 2, and similarly for the TCGA-BRCA project, the power exceeds 80% for replicates numbers as low as 66 replicates, independent of the chosen FDR threshold. Also, the differences in power for the different FDR thresholds decreases very fast with increased effect size or replicate number. However, if we take a look at the actual power for each gene, we can see some differences. In figure 4, we can see the distribution of gene-specific power split into 3 different expression levels of low, medium, and high, and 3 different replicate numbers, which are specified on the sides of the plots. On the left side are results for the drug-treated cell line data, and on the right are the data for the TCGA BRCA project. The histograms show the high dependency of the power on the expression level. The power for high expressed genes is often very close to 1 for very low replicate numbers, when the average power for that condition hasn't even passed the 80% threshold according to figure 3. The increase in power seems to come mostly from low expressed genes, which mostly have a power of 0 for the lowest shown replicate number and then slowly increase for higher replicate numbers. This can have an impact on the analysis of the results because the basal expression level of genes is not random. Therefore, genes with known low basal expression are less likely to be detected as differentially expressed. Furthermore, in figure 5, you can see the gene-specific power for genes selected for different expression levels. The figure is based on the drug-treated cell line data. As you can see, the power increase is stable and similar to the average power curve. The genes with the lowest expression level need the highest replicate number to reach a power of 80%. Overall, we can state that the implemented smooth bootstrap method allows reliable type 2 error or power analysis for RNA-seq data without the need of data simulation. Also, the estimated power is higher than expected, even for highly variable human cancer samples. The power for medium and high expressed genes is often higher than 80%, even for very low replication numbers, with 3 for the drug-treated cell line data and 54 for the TCGA BRCA project. The next steps are to implement the variance preservation during sampling and to test other bandwidth estimation methods, and finally put it all together into an R package. With that, I want to conclude my presentation. Thank you for listening.